Hello. So we'll start with the basics of hematology. The purpose of this series is to make pathology a little easier for others to understand and to get an understanding of the basics, and then we'll go on to the more advanced aspect of things. But first we'll be touching only the basics so that it's easier for everybody to understand and learn. So where do these RBCs come from? The RBCs originate from the stem cells that are present in the bone marrow, and the process of formation of RBCs is called hematopoiesis. So these stem cells, they are present in niches in the bone marrow. And what is a niche? It is nothing but a collection of cells. And these niches are surrounded by non-hematopoietic cells as well, like the supporting stroma, macrophages, fibroblast, and many other components of the bone marrow. So there are stem cell niches and the surrounding bone marrow normal cells. The stroma is also there, macrophages are also there, fibroblasts are also there, and all the other cells like fat cells are also there. So they are a collection of cells surrounded by the other normal components of a bone marrow. So it originates from the bone marrow. The RBC production, the process starts in the bone marrow. So this is a basic flow chart to make the understanding a little easier. So first of all, we have a stem cell. This stem cell originally is not committed to any series or to the production of any specific cell. Then it can either get converted into a lymphoid progenitor cell. Progenitor is like a purvaj from where it originates, the initial making cell of it, the parent cell. So it either differentiates into the lymphoid progenitor cells that is committed to the production of the lymphoid component, and the myeloid progenitor cell that is committed to the formation of the myeloid series of cells, and other cells as well like the RBCs and platelets. So first we have the lymphoid progenitor cell and the myeloid progenitor cells, and then they divide into multiple committed stem cells. Why are we using the word committed? Because these are only responsible for the production of the cells of their own lineage. So the lymphoid cell cannot make a myeloid cell. So from the lymphoid progenitor cell, we have mature T lymphocytes, B lymphocytes, and natural killer cells, and the myeloid progenitor cells. Further, they differentiate into monocytes, neutrophils, RBCs, platelets, basophils, and eosinophils. And these cells can be morphologically be differentiated and recognized in the bone marrow itself. So if you talk about the report of a normal CBC, so we have different kind of differential. When we do the differential count, we have different kind of WBCs. So some of the WBCs come from the myeloid progenitor cells, like the monocytes, neutrophils, basophils, eosinophils, and the lymphocytes. They come from the lymphoid progenitor cells. And then we have RBCs and platelets. So let's get a basic understanding of what all components are there to a bone marrow. First, we have the cellular elements and we have the stroma. The stroma is basically the supporting tissue. So we, in the cellular components, there are erythroid precursors. They are committed to the production of RBCs, myeloid precursors that are committed to the production of the myeloid series of cells, megakaryocytes and thrombopoiesis. Megakaryocytes further lead to the formation of platelets, lymphocytes, macrophages, mast cells, and plasma cells. So these are the cellular components, and the entire structure of the bone marrow is supported by the stromal component. For that, we have the stromal cells, fat cells, the blood vessels, fibroblasts, endothelial cells, iron deposits, and osteoblasts and osteoclasts. So if we proceed further and try to understand what a niche is, HSC is hematopoietic stem cell. So we are talking about hematopoietic stem cell niche. So there are two type of niches. One is the osteoblastic or end osteal niche, and the other one is the vascular niche. Why are we using these words? So, as you can understand by the name itself, osteoblastic or end osteal. So it is more close to the endosteal part of the bone to the osteoblast. It's towards the periphery of the bone of the trabecular bone, and the vascular niche is relatively more centrally placed, where it is around the blood vessels and the microvasculature of the bone. So there are two types of niche, osteoblast and vascular niche, and both of them have different functions. The stem cells, they keep going to and fro between the two niches. And osteoblastic niche, basically it is a home for more dormant kind of stem cells, and vascular niche has more active stem cells. So further, we'll be studying about these two niches, and we will get a picture also in which you can understand the relation between the two niches, and you will get the location of the two as well. So this is a very simple illustration depicting the different kind of niches. So one is the endosteal niche and this is the vascular niche. So the bone and the osteoblasts are more close to the endosteal niche. This is the peripheral part of the bone, and the osteoblasts are close to the endosteal niche, and then there is the most central part of the bone towards the center of the bone where the blood vessels are there, the microvasculature of the bone is there you find the vascular niche. And the stem cells, they shuffle depending upon the requirement, they go to and fro between the two niche. So let's summarize what we've just read about the two niche. One is the osteoblastic niche or the endosteal niche. As you can understand from the name itself, it is towards the trabecular lining of the bone. It is adjacent to the osteoblast. They are a home to the dormant hematopoietic stem cell, and they have an important role for the regeneration, and they express osteopontin at the end osteal surface. What is osteopontin? Osteopontin is a negative regulator of proliferation of these cells. So when it is not required by the body, when we don't need more hematopoietic stem cells to proliferate, what happens is that osteopontin gives a negative stimulation to the cells to stop proliferation. So when the work of these stem cells are over, not over but not required at that particular point of time, osteopontin, being a negative proliferation regulator, it halts the process of proliferation and regeneration. The other niche is the vascular niche, and it is more centrally located in the marrow, and they surround the blood vessels and the marrow sinusoids. And the hematopoietic stem cells are CD34 positive. So the marker for stem cells is CD34. It is important for mobilization, differentiation, and homing of stem cells. So, to make your understanding a little easier, so the dormant stem cells are present in the osteoblastic or the endosteal niche, and the more active and differentiating stem cells are present in the vascular niche. So when there is a stimulus, the vascular niche, the cells over there, they proliferate, and since they are closer to the marrow sinusoids and the blood vessels, they get into the circulation, they solve their purpose, they get into the circulation, proliferate, and then after the purpose is solved, they go back to the endosteal niche. So vascular niche has active hematopoietic stem cell, and the endosteal niche has dormant hematopoietic stem cells. The vascular niche being closer to the blood vessels, the stem cells can easily go into the circulation from here. So right now we used a new term, that is homing of the stem cells. What is homing of the stem cells? It is the process of mobilization of the hematopoietic stem cells from the bone marrow into the circulation and back into the bone marrow. It is called homing. So homing is just like going home, homing of stem cells. So in bone marrow transplantation, when we inject the stem cells that are cross-matched, they go and they get recognized by the microvasculature of the two types of niches, and then they go transendothelially and they get lodged into the hematopoietic spaces, and they become a natural part of the two niches. What happens is that in the time of stress, the dormant hematopoietic stem cells in the endosteal niche, they migrate to the vascular niche, And in the vascular niche, they undergo hematopoietic regeneration and myeloablation. And from there, they go into the circulation and then they re enter into the bone marrow and restore the hematopoietic stem cell pool. So another important component of the bone marrow niche is the mesenchymal stem cell. They are multipotent. That means that they have the capacity to divide into a variety of cells. They form chondroblast, osteoblast, fat cells, muscle cells, fibroblasts, and endothelial cells. So they have differentiated into these variety of cells, and they express CXCL12, VCAM1, and osteopontin. Further elaborating the function of these mesenchymal stem cells, they support hematopoiesis via causing interaction between the hematopoietic stem cells and the progenitor cells. They cause the release of soluble factors like WNT and slit proteins. They induce angiogenesis, that is production of new blood vessels. They release a variety of cytokines. They interact with tumor cells, and by interacting them, they can promote or they can inhibit the growth of a tumor or metastasis. They protect the hematopoietic stem cell niches, and they modulate the activity of various immune cells. To cover the topic of pathways and factors that regulate hematopoiesis, it's the parathormone or parathormone receptor P signaling pathway, WNT and beta catenin signaling pathways, the sympathetic nervous system, Type 2 and OG1 signaling pathway, Notch signaling pathway, and Hedgehog signaling pathway. Now, this is an entire topic on its own, and it's not relevant to elaborate it over here because it would get the topic complicated. But just to complete the list, these are some pathways that we can study about later, but there is no relevance of elaborating on this any further. Further elaborating more growth factors that are responsible for hematopoiesis, as you can see, there are a variety of them. We don't need to go into the details of each and every one of them. It's more of a theoretical topic, but the most important here is erythropoietin that we've already spoken about. And you can go into the details by just going through this table. No more detail is required. Further elaborating the sites of hematopoiesis, in the embryo stage, up to the third week, the yolk sac is responsible for hematopoiesis, and up to the third month, the liver is responsible. Then, in the fetal life, fourth month onwards, the role is taken up by the bone marrow. And if you pay attention, then at 18 years and above of age, vertebra, ribs, sternum, skull, pelvis, proximal epiphyseal region of the humerus and femur are the main sites of production of RBCs. So the bone marrow is the main source of production, these being the primary sites, and in the embryo stage, it's yolk sac and liver, and from the fetal stage, fourth month onwards, we have the main load of RBC production on the bone marrow. So in the bone marrow, there are multiple pluripotent stem cells that get influenced by multiple cytokines, and then they become committed to the erythroid series, now being called the erythroid progenitor cells. These erythroid progenitor cells are present freely in the circulation, and while they are in the circulation at this stage, they acquire receptors to erythropoietin. Erythropoietin is produced by the kidney. And by the effect of erythropoietin, these erythroid progenitor cells further differentiate into various stages of the erythroid series. We'll elaborate the multiple stages further in the presentation. But in the absence of this erythropoietin, what happens to these progenitor cells if erythropoietin is absent? These cells, they automatically undergo apoptosis, that is programmed cell death. So erythropoietin acts on these erythropoietin receptors present on erythroid progenitor cells that are freely present in the circulation. So what happens is that erythropoietin, as discussed earlier, being produced by the kidney, its production is stimulated in the case of hypoxia. So in hypoxic stress, the body needs more oxygen. So what happens is the erythropoietin volume is increased. Erythropoietin goes and acts on the receptors that is present on the surface of the erythroid cells, and it stimulates them to further proliferate and differentiate. So erythropoietin causes the increase of RBC production via acting on these erythroid series cells. So, talking about the first stage of the erythroid series, it's the proerythroblast. It is one of the largest cells that is seen in the circulation, and it ranges from up to 12 to 20 microns. So, as you can see in this image of a bone marrow aspirate, this is by far the largest cell that you can visualize. You can see this is the nucleus, and it has a stippled nuclear chromatin, and there are one to two nuclei that are barely visible. The cytoplasm of the cell is basophilic, and this cell is highly metabolically active because of the large amount of mRNA. And the main purpose of this mRNA is to manufacture a number of proteins. So, the first stage of the erythroid series is the proerythroblast. So, after the first stage of a proerythroblast, the next is an early erythroblast. As you can compare from the previous image, the size of the cell is relatively smaller. So it's 12 to 16 microns, and the nuclear chromatin becomes relatively more condensed. So as you progress in the stages of the erythrocyte series, the size decreases, the nucleus becomes more condensed, and gradually the nucleus disappears. So in this, you can see from the previous stage, the cell size is smaller, the nucleus is more condensed, and the cytoplasm is light basophilic. Initially, in the previous stage, it was more basophilic. In this stage, it is lightly basophilic. And the nucleoli that were 1 to 2 previously are lost. There are no more nucleoli that can be seen in this stage. After the early erythroblast, there comes the next stage of an intermediate erythroblast followed by a late erythroblast. So the one that is being pointed by the red arrow is the intermediate erythroblast, and the ones being pointed by the black arrow are the late erythroblast. So comparatively, the size again reduces, and it becomes initially that was from 12 to 16 microns. Now it's reduced further down to 10 to 12 microns. The cytoplasm starts gaining a relatively more reddish color. It gets a reddish tinge, that is the polychromatophilic color. This is because the hemoglobin production has started, and the nucleus becomes more condensed. So followed by the stage of the intermediate erythroblast, we have the late erythroblast. And in this particular image, again, you can see both the stages, and you can compare the two. So the size has reduced, the cell size is reduced. The cytoplasm is more red in the late erythroblast. The nucleus is more condensed and smaller. And when the body needs more RBCs, the bone marrow gets into a condition of stress. In this, the RBC production is increased. And under a condition of stress, these cells, the late erythroblast, can at times be seen in the circulation. Gradually, what happens is this late erythroblast, it passes through the spleen, and the nucleus gets extruded, and it forms a reticulocyte after that. It's negatively staining zone. It has a negatively staining zone that is called the Golgi zone, and this can differentiate from this from a lymphocyte. This at times can resemble a lymphocyte, so the negatively staining zone can differentiate this from a lymphocyte. So the next stage is the reticulocyte stain, and in the Romanowski stain, this cell, it appears a little more bluer than the normal RBCs, and the nucleus in this stage has been removed on the very first day of production, and these can be detected via the supravital stains. As you can see that there are two types of reticulocytes that you can see. This one is an early reticulocyte in which you can see the bluish dots more condensed, and gradually they disappear in the late reticulocyte stage when they are progressing to form the mature RBCs. So in situations where the erythroid activity is stimulated, like in case of hemolytic anemia, iron deficiency, sorry, iron therapy or folic acid therapy or vitamin B12 deficiency, there is an increase in the count of the reticulocytes, and there is a significant reduction in the size of reticulocyte, that is below 0.5 percentage in aplastic anemia and red cell aplasia. So this can be seen by supravital stains like reticulin. So finally comes a mature RBC. It has a lifespan, as we all know, of 100 to 120 days. It is anucleate. So by this stage, the nucleus is entirely lost. Even the reticulocyte doesn't have a nucleus. But in this, you can very clearly see that there is no nucleus. And the size of a normal RBC, of a normocytic normochromic RBC, is that of the nucleus of a small lymphocyte. So when we do a GBP or a peripheral blood smear reporting, we always try to compare the size of an RBC with a lymphocyte because we want to compare the nucleus. And in the RBC, we can all see that there is a central one-third pallor followed by a two-third darker shade rim. This is because, as we know, the RBCs are biconcave and the thickness of the plate is less in the center as compared to the periphery. The function of the RBCs is to carry oxygen and carbon dioxide to and fro from the body. So, summarizing the various stages of the erythroid series, first we have the pro-erythroblast followed by an early erythroblast, then the intermediate erythroblast, then the late erythroblast followed by the reticulocyte, and finally the mature RBC. Other general points that we need to know are that an adult body produces about 3 into 10 to the power 6 per liter RBCs per day. And the nutrients that are very important for the production of RBCs are iron, vitamin B12, and folic acid. Other nutrients that are not as important are vitamin B6, that is a coenzyme involved in the metabolism of various amino acids, cobalt, that is an essential trace element as a part of vitamin B12, that is cyanocobalamin. Vitamin C, that reduces the ferric iron to the ferrous form so that it can be absorbed by the body, and vitamin C and copper, that is ceruloplasmin like substance which transfers iron from cells to plasma.